A cell's secrets can be divulged by its surface, decorated with tens to hundreds of thousands of molecules that help immune cells determine friend from foe. Some of those protruding molecules are antigens that trigger the immune system to attack, but it can be difficult for scientists to identify those antigens, which often vary across individuals, in the molecular forest. A team of Stanford scientists led by Polly Fordyce, an institute scholar at Sarafan ChEM-H, has developed a new method to faster and more accurately predict which antigens will lead to a strong immune response. Their approach, which was reported in Nature Methods on September 5, could help scientists develop more effective cancer immunotherapies. T cells, a class of immune cells, crawl along and squish past other cells as they patrol the body, using T cell receptors to molecularly read peptides, or short pieces of proteins, which are cradled within larger proteins called major histocompatibility complexes, PMHCs, that project from cell surf. Healthy host cells display an array of PMHCs that do not trigger an immune response, but once T cells recognize disease-indicating peptides, they become activated to find and kill cells bearing these foreign signatures. Understanding how T cells sensitively distinguish these antigenic peptides from host peptides to avoid mistakenly killing host cells has long been a mystery. A T cell can detect a single antigenic peptide amongst a sea of 10,000 or 100,000 non-antigenic peptides being displayed on cell surfaces, said Fordyce, assistant professor of bioengineering and of genetics. But sometimes, it has the opposite effect. It's kind of like a Chinese finger trap, said Fordyce. When you pull a bit at the receptor-antigen interaction, the binding actually lasts longer. Cellular mimicry identifying the best antigen-receptor pairs requires simultaneously applying that sliding, or shear, force between a peptide and a T cell and measuring T cell activation, ideally thousands. The study's first author, postdoctoral scholar Yinian Feng, developed a trick that allows the team to measure 20 unique peptides interacting with thousands of T cells in less than 5 hours. After depositing a T cell atop each bead and waiting long enough for receptors to bind to the peptides, they then very slightly heated the bead. The bead's expansion increases the distance between tether points, and the corresponding stretching of the T cell mimics the force it would experience sliding along cells in the body. After exerting that force, the team then measured how active the T cells were. They could do hundreds of individual experiments in parallel by using beads that are each labeled with a unique color, making it possible to track multiple different PMHCs. They took 2 sets of pictures tiling across each slide after each run. One set that tells them which PMHC a given bead is displaying and another that tells them how active each T cell atop that bead is. Thanks for watching. Please subscribe my channel for more videos.